Good morning friends. My name is Nipin Tiwari and you are watching an educational program called Learn at Your Own Pace. In this educational program, you have the facility to learn the things at the convenience of your home. At the same time, you can go from one slide to another slide, pause, ponder, return back, and in this way you will be able to learn the things at your own speed. If you have any doubt, you can once again refer and learn. In today's presentation, we will discuss sexual reproduction in flowering plants. This is the third part of our chapter number 2 of class 12 biology. Before we start this discussion, I would like to tell you that this content has been developed by me for the welfare of students community so that they could learn at the comfort of their home. But at the same time, I would like to remind that this content should not be used for any commercial purpose. So far as the references are concerned, I have referred the NCERT textbook and I have taken the images from NCERT as well as the website google.com. In today's discussion, we will learn the process of double fertilization in angiosperms. We will also learn the post-fertilization events, that is, development of endosperm and development of embryo. We will also learn about the structure of a dicot and a monocot embryo. Then we will learn about the development of a seed. At the end of our discussion, we will also learn about the differences between an albuminous and non-albuminous seeds. So let's start our discussion with the process of fertilization in angiosperms. Students, you can see that this is a well-labeled diagram of a female sex organ in angiosperm. The carpel or pistil, it consists of the stigma. The tip of the carpel or pistil is called stigma, and the long or tubular style, and at the base you will find a flask-shaped ovary. So what happens during fertilization? You will find that the pollen grains have landed over the stigma and the pollen tubes are growing in the downward direction. So this is the ovary, and inside the ovary you will find the ovule, and inside the ovule you will find a well-developed embryo sac. So here in this picture I have tried to show the well-developed embryo sac, where you can find two synergids. This is one synergid and another synergid is lying behind this one, and this is one egg cell, and this is the central cell, and at the central cell you can find two polar nuclei. These are the two polar nuclei. This end is called micropylar end, and this end of the embryo sac is called chalazal end. At the chalazal end you can see the three synergids. In the central cell you are finding two polar nuclei, and at the micropylar end you will find the egg apparatus The egg apparatus will consist of two synergids and one egg cell. In the synergids, what you will find is the finger-like projections called filiform apparatus. So, here I have tried to show the process of I have tried to show the process of fertilization in angiospermic flower. So in this magnified diagram of embryo sac, you can observe the growth of the pollen tube through the micropylar end. This is the micropylar end. Okay. This is the micropylar end, and at the micropylar end you can see the growth of the pollen tube. At the micropylar end you will see the growth of the pollen tube, and where the pollen tube is entering into, the pollen tube has two male gametes and it is releasing its contents, that is the two male gametes, into the synergids. This cell is called synergid, and you will find here special finger-like projections, finger-like structures. These structures are called filiform apparatus, and the filiform apparatus, they in fact guide the two male nuclei, so that one male nucleus will be guided by the filiform apparatus to enter into this egg cell. This is the egg cell. Egg cell, the number of chromosomes in the egg cell is N. One set of chromosomes, that is called haploid. Another N number of chromosomes, that is another set of chromosome, is carried by the one male gamete. So the first male nucleus or the male gamete will enter into the egg cell, and these two, the egg nuclei and the male gamete, will fuse to form a 2N. That will form a zygote. Zygote will be 2N. That will be a diploid condition. What will happen to the second male gamete? The second male gamete will be guided by the filiform apparatus, and it will reach into the central cell. Inside the central cell, you will find two polar nuclei. Each polar nuclei is also having one set of chromosome, that is N. So these two polar nuclei and this second male gamete, that will fuse together to form a 3N, that is called triploid primary endosperm nucleus. Okay, and this is taking place inside the central cell. Now, what are the two prominent events which are taking place inside the embryo sac? These two prominent events are the fertilization. That fertilization is better known as double fertilization. What is double fertilization? Double fertilization means syngamy and triple fusion. Now what is syngamy? Syngamy means fusion of the one male gamete, that is the first male gamete, with the egg cell to produce a diploid zygote. This is called syngamy. And what will happen to the second male nucleus? As I said earlier, the second male gamete will reach inside the central cell, and it will fuse with the two polar nuclei to form a triploid primary endosperm nucleus. Since in the central cell, the three haploid nuclei are fusing together, so we will call it triple fusion. That means fusion of three haploid nuclei. Now, fusion of the male and female gametes is called syngamy. Fusion of two polar nuclei and the second male gamete is called triple fusion. How many fertilizations are taking place inside this embryo sac? Two fertilizations. One fertilization is called syngamy and another fertilization is called triple fusion. The result of syngamy is the diploid zygote, and the result of triple fusion is the primary endosperm nucleus. This phenomenon is called double fertilization. So here with this flow diagram you can see that double fertilization in an angiosperm consists of two processes. One is called syngamy and another one is called triple fusion. What is syngamy? Fusion of the first male gamete with the egg cell, and the triple fusion is the fusion of the second male gamete with the two polar nuclei. So what is the significance of double fertilization? The significance of double fertilization is that the zygote, it develops into embryo, which is a diploid structure. We call it 2N. And the second important function of double fertilization is formation of PEN, primary endosperm nucleus. It is a triploid structure. The number of chromosomes is 3N and it develops into a triploid structure endosperm. It is a nutritive tissue which will nourish the developing embryo. So in this way we could discuss the double fertilization and the triple fusion. Now fertilization has taken place. After fertilization, what are the events and what are the structures which are formed inside the embryo sac? All those structures are called as post-fertilization structures, and the events are called as post-fertilization events. Now, here we will find that the primary endosperm nucleus develops into endosperm after the process of fertilization. The zygote, it will develop into a full-fledged embryo. The ovule, which was there inside the ovary, it will develop into a seed, and the ovary itself will develop into a fruit. So here one question which is repeatedly asked in the board exams, CBSE exams, that the endosperm development precedes the embryo development. Why? What does it mean? The question means that in reality what happens during the formation of post-fertilization structures, these polar nuclei, they will produce a primary endosperm nucleus, and this primary endosperm nucleus, it will divide and redivide and fill this entire space of the central cell into endosperm, a nutritive tissue. Okay, and then only the zygote will be dividing and it will finally give rise to a well-developed embryo. So here the question is, why the endosperm development takes place first and then the embryo development takes place later on? Okay, we will learn about this concept a little later. The things will be more clear to you in the next few slides. So in the previous slide we saw that in the central cell there is a triploid primary endosperm nucleus. It will divide and redivide into a full-fledged nutritive tissue that is called endosperm. So depending upon the process by which the endosperm or the nutritive tissue develops, we can see that there are three different types. One is called the nuclear type of endosperm development. This is the most common type. The second one is called the cellular type of endosperm development, and the third one is somewhere in between or intermediate of the nuclear and the cellular type. This is known as the halobial type of endosperm development. So in the nuclear type of endosperm development, you can see that this is the primary embryo sac, and this cell nucleus placed in the central cell is called primary endosperm nucleus. It is also known as PEN. So this PEN, it will undergo the first mitotic division and it will form the two nuclei. This is the first one and this is the second one. And after that, it will continuously divide and redivide to produce a number of nuclei. So all these are the nuclei and they have been pushed to the peripheral region of this central cell or the embryo sac. So this area is occupied by the vacuole. But later on, this entire central cell is filled with this free nuclei, or this condition is called as free nuclear division. And in this way you will find the many nuclei in the central cell. In this diagram, the wall formation has not yet taken place. But when you go to the next diagram, here you will find that the peripheral area or the outer area of the embryo sac, it is having the cell wall formation. In this area you can see the cell wall formation has taken place. And gradually this cell wall formation will proceed towards the center area, and here also this area will also be occupied by cells having the cell wall. So in this way you could see that the primary endosperm nucleus here, it has divided and redivided into a number of nuclei, and then at the end of this process the cell wall formation has taken place in the centripetal manner. Centripetal manner means from the peripheral area to the central area. So this is called as the nuclear endosperm. This is the most, as I said in the earlier slide, this is the most common type of endosperm development. And the examples are the cotton, maize, wheat, Capsella bursa pastoris, etc. In the second type, that is called cellular type of endosperm development, you will see that the primary endosperm nucleus is there in the central cell. So as soon as this primary endosperm nucleus divides into two daughter nuclei, a wall formation has taken place. Now you can see the two daughter cells, and the further nuclear division will simultaneously be followed by the wall formation. So here you will never find a free nuclear condition. You will find that as soon as a nuclear division has taken place, wall formation will take place immediately. So in this picture you can see that of course there are nuclei which has not been shown in this diagram for the sake of convenience, but as soon as the nuclear division has taken place, cell wall formation has also taken place simultaneously. So this type of endosperm development is called cellular type of endosperm development. Now here you can see that this entire the central cell or the embryo sac is occupied by the cellular endosperm, and this dark area is actually the developing embryo. So this type of embryo development is seen in grasses, monocots, barley. Then you will also find the cellular type of endosperm development in petunia and datura, balsam, etc. Now in our textbook there is one case of coconut endosperm. It is very interesting, and we all are aware that a coconut has the outermost green part that is called pericarp. The pericarp has three regions, that exocarp, mesocarp and endocarp. And when this is the endocarp is the hardest layer, very tough and hard like stone, and inside the endocarp most of you are interested in this cellular portion, that is white portion, which I am showing now by yellow. This white portion is actually the coconut meat, and this is the solid endosperm or the cellular endosperm. And then after consuming this coconut meat or gadi or kachi gadi, you will find that inside this, the central area is occupied by the liquid endosperm. This liquid endosperm is used as a refreshing coconut milk. It is a refreshing and nutritious drink. So the coconut has two types of endosperm simultaneously. The centralmost area is occupied by the liquid, which is in the form of a water, the coconut water. This is the liquid endosperm, where you will find a number of free nuclei, and it is surrounded from all the sides by white colored endosperm where cell wall formation has taken place. Okay, so this is the cellular endosperm, or it is also called as the coconut meat. Then the third type of endosperm development is the halobial endosperm development, which is somewhere between the cellular type and the nuclear type. How it is of the intermediate type of development, that will become clear by this help of this diagram. So this is the primary endosperm nucleus. This nucleus is called the primary endosperm nucleus. The primary endosperm nucleus is dividing here into two daughter nuclei, and one daughter nuclei is surrounded by a cell wall at the chalajal end. So this is the cellular endosperm, and this is the nuclear endosperm. This is the larger one, which is toward the micropillar pole of the embryo sac. So this cellular endosperm will remain as it is, and the another one which is occupying the most of the part of the endosperm, it will develop in the manner in which the nuclear endosperm has development of nuclear type of endosperm has taken place. So ultimately this nucleus, the free nuclei, they will also once again be followed by cell wall formation, which will definitely proceed from the peripheral area toward the central area, that is in the centripetal manner. And finally you will find the endosperm or nutritive tissue, and again this darker area is the developing embryo. So this type of endosperm development, that is halobial type of endosperm development, it takes place in a number of monocotyledonous plants. In plants in which the embryo has only one cotyledon. Now we have seen that the endosperm development has already taken place, and now endosperm development is full of nutritive tissue. It has accumulated a lot of nutritious material so that it could nourish the developing embryo. So the zygote, which was a diploid structure formed as a result of the syngamy or fertilization, now it is the time for the zygote to develop into an embryo, and this development will take place at the micropillar end of the embryo sac. So now it is clear that most of the zygotes, they divide and develop into a well developed embryo only after certain amount of endosperm has already formed. Why? Because This is an adaptation to provide assured nutrition to the developing embryo, so that when the embryo is undergoing development, it is supplied with uninterrupted nutrients, or it ensures the uninterrupted supply of nutrients to the developing embryo. So the zygote will divide and redivide to form a proembryo, and the latter developments will witness the structures like globular, heart-shaped, and a matured embryo. So here we find in a board exam, we find the question like, with the help of label diagrams, represent the different stages of embryogeny in a dicot plant or in a dicot embryo. So it was clear from the earlier discussion that this is the embryo sac, and inside the embryo sac, now this is the zygote, this is the zygote 2n, okay, and this is the central cell where there was a triploid 3n primary endosperm nucleus PEN. Now in our earlier discussion, we already know that this entire area is now occupied with the endosperm or the nutritive tissue. Once this entire area has been occupied with the nutritive tissue called endosperm, now it is the time for this diploid structure, the zygote, which is having n number of chromosomes, 2 sets of chromosomes. Now it will undergo division. So this is the zygote. The first cell division will be the transverse division or horizontal division, and this is called a two-celled proembryo stage. This is a two-cell proembryo stage. The two cells are, one is called basal cell and another one is called terminal cell. This basal cell, let me take the pen. So this basal cell, it will produce a structure called suspensor. This structure is called suspensor. So this structure has been produced by the basal cell. What will happen with this terminal cell? Suppose this is a terminal cell, okay? I'm drawing a diagram. Now this terminal cell will undergo our horizontal division. It will become two-celled stage. Then it will undergo a longitudinal division or vertical division. So what you can see? You can see the 4 daughter cells. Then it will undergo a periclinal division. This type of division is called as the periclinal division. Now how many cells? 8 cells. And then it will undergo anticlinal divisions. And then with the help of all such divisions, I repeat again, vertical division, that is called longitudinal division, transverse, anticlinal division, periclinal division, all such will lead into the formation of the embryo. This is globular embryo, which you can see here. Globular, which has a spherical structure. So from where this spherical structure has been formed, this is the terminal cell. You must remember, in the medical entrance exams, it is asked that globular embryo, it originates from basal cell, basal cell or from terminal cell or from basal cell and terminal, or there may be one choice called either from basal cell or from a terminal cell. It varies from species to species. Students, you must remember that the basal cell will never form a part of the embryo. The basal cell will form only the suspensor part, and the terminal cell, it is responsible for the development of this part of the embryo or the embryo proper. Now the next stage of the embryo development is the heart-shaped embryo, in which you can find that the embryo has somewhat occupied the shape of a heart. So this is called the heart-shaped embryo development. Okay. Now in the next slide we can see that now the embryo has the fully developed structure, mature dicot embryo, where you can find this structure called suspensor, and this is the embryo proper. In the embryo proper you can find structures. This diagram is called cellular diagram, where the cellular details has been shown, and this diagram is called the line diagram. In exams you often find such questions in 2 marks, that draw a well-labeled diagram of a matured or fully developed dicot embryo. So you must draw this diagram, which is also shown in the NCERT textbook, that this dicot embryo has this structure which I am showing right now. This structure is called embryonal axis. This embryonal axis has the tip of the embryonal axis is called, let me take once again the highlighter. So the tip area is called plumule. This is called plumule. This is the tip of the embryonal axis. And again, this area is occupied by the tip. This is called radicle. This plumule will grow into a shoot, and this radicle will grow later on into root. Since the radicle will develop into root, and the roots, they are subjected to mechanical injuries when they penetrate across the different layers of the soil, there must be a protective mechanism. So the radicle, they are not naked. They are surrounded, or they are guarded by or protected by a root cap. Okay. So the embryonal axis has structures like the plumule. This structure is called embryonal axis, plumule. Okay. Then radicle, and the radicle or the root tip, it is occupied by a protective structure called root cap. And this is the point. Again, let me select the pen. This is the point. This is the point where this leaf-like structures are attached to the embryo proper. This is the embryo proper. This structure is the embryo proper, or we are calling it the embryonal axis. Now this structure you can see, this is a leaf-like structure. This is called cotyledon. So in this diagram we can see two cotyledons. These are the two cotyledons. These two cotyledons are attached to the embryonal axis at one point. Okay. So the part of the embryonal axis which is just above the point of attachment of the two cotyledons, this part is called epicotyle. This part is called epicotyle. Again, I told you, this is the place where these two structures, the two cotyledons, are attached to the embryo proper. So the part of the embryonal axis below this point of attachment is called hypocotyle, which has been shown here. We have few more diagrams where this structure will be clear. So I told you that what kind of structure the dicot embryo have. They have one embryonal axis. This is the embryonal axis, okay, and the tip of the embryonal axis here you will find structure called plumule, and then lowermost part of the embryonal axis is occupied by a structure called radicle. Radicle is covered by root cap. This is the point of attachment. This is the point of attachment of the cotyledon to the embryonal axis. So the area of the embryonal axis which is above this point of attachment is called epicotyle, and the area of the embryonal axis which is below this point of attachment of cotyledon is called hypocotyle. Now we also have the structure called monocot embryo. In a monocot embryo, you will find a structure like scutellum. Scutellum is in fact the rudimentary cotyledon. In the dicot seed, you will find the two cotyledons, but in monocot seed, we will find only one cotyledon, and that too in a very rudimentary manner, in a very small scale. So that's why such rudimentary cotyledon, that single cotyledon in monocotyledonous seeds, are called scutellum. And in a monocot, you will find that this is the shoot apex. This area is called the embryonal axis. This is the embryonal axis. So this is the shoot apex, okay, and the shoot apex is covered by a leaf-like structure. This is called coleoptile. This is called coleoptile. And the lower part of the embryo is called the radicle, and radicle is protected by a root cap, and this root cap, it is also surrounded by a leaf-like structure called coleorhiza. So these are the two additional structures. This is the embryonal axis in monocot seed. This is the embryonal axis in a monocot seed. And monocot seed has the shoot apex or plumule and the root apex called radicle. And there is no doubt a root cap. But the shoot apex is protected by a leaf-like structure called coleoptile, and the radicle and root cap, they are protected by a leaf-like structure called coleorhiza. Okay. Now we have studied in detail about the manner in which endosperm development takes place. Then we also studied how the embryo development takes place in angiosperms. Then we also saw that there are two types of endosperms. There are two types of embryo, monocot embryo and dicot embryo. In dicot embryo, we could see that there were two cotyledons, whereas in monocot embryo, we could see that there is only one cotyledon, and that too is very, very small and non-functional, and that is called as scutellum. Now we will study a little bit about the seed. What is a seed? Seed in fact is the most important structure which has been formed as a result of sexual reproduction in flowering plants. And if it comes about the definition, we can say that a fertilized and mature ovule is called seed. So when we go into the structural details of a seed, say for example a gram seed which has been shown in this picture, in this diagram. When you will split open a seed of gram or chana, what you will find? You will find an outermost covering called seed coat. Seed coat has two layers, testa and tegmen. The outer layer is called testa and the inner layer is called tegmen. Now let me remind you from where this seed has been formed. The seed has been formed from the ovule. So you remember that the ovule was surrounded by two layers, that is called integuments. So the two integuments, the outer layer and the inner layer, which was there in the ovule in the form of integument, now it has become, due to the hardening of the integument, it has led to the formation of these two membranous protective coating of the seed called testa and tegumen. Now in a seed, if it is a dicot, you will find two cotyledons, or if it is a monocot seed, you will find only one cotyledon, which we have already seen in the previous diagram. The cotyledons are generally thick and they are swollen. Why they are swollen? Due to storage of food reserves. And in a seed you will find the embryonal axis, that is called plumule, and plumule is also called as the future shoot because it will germinate the seed. When the seed will germinate, the plumule will give rise to shoot, and the radicle, it is also called as future root because after seed germination the radicle will give rise to the root. Now from this diagram you could see that the seed coat has been shown as testa, the tegmen will be inside it. Here this testa and tegmen are clearly seen. Then you can find that these are the two cotyledons. This is one cotyledon, this is another cotyledon, and this area has been shown by the embryonal axis. This embryonal axis has been magnified here. You can see that this is the plumule, okay, and this area is called radicle, and of course this area will be the root cap, and this entire structure is called embryonal axis. Okay, this was the point of attachment. This was the point of attachment where these two cotyledons are lying attached with this embryo proper. So this area, let me use the pen here. So you can see that this area is called epicotyle, and this area, this is the point of attachment. This is the point of attachment where the two cotyledons are attached to this embryo proper. So this area will be called as hypocotyle, and no doubt this will be radical, and this will be the root cap. So this was the structure of a seed. Now we will learn few more structures of a seed. We must not forget once again that from where this seed has come. This seed has developed from ovule. So whatever were the structures present in an ovule, little bit they will be modified and they will form a part of the seed. So we can see that in the ovule there was micropyle. So micropyle here also it remains as a small pore in the seed coat. What is the importance of micropyle? It facilitates the entry of oxygen and water during the process of seed germination. In the ovule we had found a structure like funicle. This funicle will be transformed into the stalk of the seed, and the hilum marks the point of attachment to the stalk. Okay, so now a phenomenon called seed dormancy. What is seed dormancy? Seed dormancy may be defined as a condition of seed when it fails to germinate, even though the environmental conditions usually favorable for active growth are present. It is a condition when you are providing everything which is required for the seed to germinate. You are providing oxygen, you are providing water, you are providing soil, you are providing humidity. Even then, if the seeds fail to germinate, this condition is called as seed dormancy. What happens during seed dormancy? When the seeds are mature, they dry up and the water content is reduced to 10 to 15% by mass, and the seeds, they become relatively dry. So what happens? Do the seeds, they stop their metabolic activity? No, not at all. The general metabolic activity during seed dormancy actually slows down. So the embryo, it enters into a state of inactivity. This state of inactivity or suspended animation is called seed dormancy. Now we have reached at the end of our discussion, where we can see that the seeds are of two different types. One kind of seed is called the albuminous seed or the endospermous seed, and the other one is called the non-albuminous or the XII seeds. What happens with the albuminous seeds? The albuminous seeds actually, they retain a part of the endosperm as it is not completely used up during embryo development. You know that the endosperms, they supply nutrients to the developing embryo. So when the embryo is undergoing development and it is becoming a matured embryo, mostly it will consume most of the endospermous tissue. But suppose it has not consumed up all the endospermous tissue, large part of the seed still contains endosperm. Such seeds are called albuminous seeds. Example of the albuminous seeds are wheat, maize, barley, castor and sunflower. But what happens in the non-albuminous seeds? In the non-albuminous seeds, the embryo, the developing embryo has consumed most of the endosperm. So no residual endosperm is remaining in the seed. It has been completely consumed during the embryo development. Such seeds are called non-endospermous or non-albuminous seeds, or they are also called as X-albuminous seeds. And examples of such seeds are dicotyledonous seeds, mostly the members of the pulse family, or dalhan or dal. All the pulses, you will find that the seed will have two cotyledons. Example, the prominent examples are pea and groundnut. So now it's time to sum up our discussion. In our today's discussion, we studied about the process of double fertilization. Then we also studied about the post-fertilization events, that is syngamy and triple fusion. We could see that how endosperm develops, and there are three types of endosperms: the nuclear type of endosperm development, the cellular type of endosperm development, the halobial type of endosperm development, and we also learned the type of endosperm in a coconut. Then we studied the process of embryogenesis, that is the development of an embryo. Then we discussed in detail about the structure of a dicot and a monocot embryo. We also discussed about the development of a seed. We could see the different types of dicot seed and monocot seed. And then at the end of our discussion, we could see the difference between an albuminous and a non-albuminous seeds. Thank you friends for watching this video. I hope you have got a good learning experience today. This presentation was a Kendriya Vidyalaya number 1 Bhopal presentation. Keep watching and subscribe my video for the further knowledgeable discussions. Thank you.